All right, so let's go ahead and pick up where we left off with the first part of the chapter 9 lecture. If you think about what the big lesson or the big takeaway from that first part is, of course, Mendel's famous pea plant experiment. And in that experiment, he was able to show that traits in progeny, in other words, in the F2, F1, F2, F3 generation, and so on, will pop up with in a statistically reliable pattern that shows that inheritance must be through two alleles. And this makes sense, of course, because we get one allele from our mother and one allele from our father. So Mendel was able to conclude from those experiments that we have this discrete packet of information that can't change. And that piece of information is called a gene. And we get one of those pieces from our mother and one of those pieces from our father, or the male and the female donor. I don't know why I don't like it sounds odd to call a plant a mom and a dad, but you get the idea, right? You have a female donor and a male donor of information, and what they're donating, of course, are those discrete pieces of information that we're going to call genes. Now, the other piece of information, of course, that he garnered from that with the way that the phenotype showed up was that one of those alleles or one of those traits were dominant to the other trait, which of course was then recessive. So the big conclusions, of course, that Mendel had was, of course, this idea that we have these discrete pieces of information. We get one from each of our parents, and one of those alleles will be dominant to the other. So let's go ahead and look at dominant and recessive characters in a little bit more detail. So here are some autosomal dominant traits, and there's lots and lots of these that we can point out in the human population, for example. And ask yourself, first of all, before we get started on pointing out what these are, ask yourself what autosomal means. You should know what this term means because we've seen it before. So of course, autosomal means that it's found on a chromosome that is not a sex chromosome. Remember, autosomal chromosomes are the 22 chromosomes that aren't are X and Y chromosomes. And those contained in those chromosomes are alleles that can lead to, there's thousands and thousands of them, of course, but here are some examples. So for example, we have a dominant allele can lead to a widow's peak on your forehead if you have your hair that grows in a kind of a V pattern in the middle of your forehead, attached versus unattached earlobes, freckles, the ability to roll your tongue. Try it really quick. I think I could do it. Wait, maybe I can't do it. I can do it. And then, of course, this one kind of blew. I looked at this picture for a little while before I figured out that, of course, he has 6 fingers. That's a dominant trait, which is kind of an interesting idea. And a lot of people really get this incorrect idea that a dominant trait is the one that pops up in the population the most often. And that's not actually true. Dominant trait simply means that in a heterozygous individual, the dominant trait is the one that's actually expressed. Very important difference there. Now, on the other hand, we can have dominant disorders. So these are autosomal dominant disorders as opposed to just traits. So these are disorders that lead to some, bad or not ideal outcome. And 3 examples of a dominant disorder would be achondroplasia, which is a type of dwarfism, Huntington's disease, which is a really painful, lethal disease that adults can get, and the onset, I think, starts in your late 40s. And then a cholesterol condition called hypercholesterolemia, which leads to lethally high levels of cholesterol in the individual that has this allele. And so here you can see that if we have 2 parents, one is a dwarf because he contains the dominant, he's heterozygous for the dwarf allele, and the mother is normal. She is recessive for that dwarfism or achondroplasia allele. And you can see that the sperm is going to contribute either the dominant allele or the recessive allele. And of course, the egg can only contribute that recessive allele because that's all the egg has, are the lowercase d's. And so when we drag these across, we can see that they have a 50-50 chance of having a child with achondroplasia. They have, of course, a 50-50 chance of having a normal-sized child as well. So here's one example of how a dominant disorder could work in terms of an inheritance pattern. Now, on the most genetic disorders that are most inherited genetic disorders are recessive. The individual, the offspring has to be homozygous recessive in order to have that trait expressed. So these three, achondroplasia, Huntington's disease, and hypercholesterolemia, are three exceptions to this rule. Typically, these genetic disorders are recessive in nature. A few examples of recessive inherited disorder would be cystic fibrosis, Tay-Sachs disease, and the metabolic disease PKU. Another example is albinism. It's not really, I guess I'm not totally clear on this, but I don't think this is really a disorder. I don't think there's any illness or any necessarily drawback to having albinism, of course, except that you're probably much more susceptible to getting sunburn because you basically have no melanin at all in your skin. So I would consider that pretty inconvenient for sure. So let's look at what happens if we have two parents that are normal in phenotype. In other words, they have normal pigmentation. However, they're both carriers for the trait. Notice that they're both heterozygous for albinism. So if these two parents were to have children, they have a 1 in 4, a 25% chance of having a child with albinism or an albino child. They have a 25% chance of having a homozygous dominant normal child, and they have a 2 in 4, right, 50% chance of having a child that's a carrier for albinism. So again, remember this is if you haven't done the the chapter 10? Yeah, chapter 10 lab. Yeah, we do these Punnett squares. We do lots and lots of examples of these Punnett squares. So you'll get some practice in doing these in that lab if you haven't done it yet. All right, so let's go over some terminology. I think I've mentioned some of these words already, but I just kind of want to formally describe what these words mean. Remember that we have here two chromosomes, and we know that are they duplicated or unduplicated? Ask yourself, are these two chromosomes duplicated or unduplicated? Hopefully you guess that they are unduplicated because they don't have sister chromatids, right? They're in that I shape, and here's the centromere here. And remember that we can always count chromosomes by the number of centromeres that you see. We see 2 centromeres, 1 here and 1 here. And we know that these two chromosomes are homologous to one another because they have the same genes in the same location. Remember, loci or locus is a fancy word for location. They have the same genes in the same location on these chromosomes. So this is, for example, I think we're using the red one for mom. This is our female donor, the chromosome that we got from our mother. This is the chromosome that we got from our father. Now, ask yourself, just by looking at these chromosomes, can you figure out which is the dominant allele and which is the recessive allele? Remember that we can always tell dominance and recessive, at least if we see the symbolism, by the capitalization. So if the allele is capitalized, in the case of the P here and B here, we know that that's a dominant form of that allele, whereas if it's lowercase, we know that that's a recessive allele. So here we can see that the genotype for this particular set of chromosomes is that it's homozygous dominant for whatever this P allele stands for. Homozygous dominant because they're the same. Remember, homozygous means the same. And because it's capital, we know that it's homozygous dominant. Now these two, on the other hand, are both recessive, so it's the same. So we're going to call this group, or this allele, these alleles, homozygous recessive. And then finally, because we have two different alleles, remember hetero means other or different, so we're going to call these alleles heterozygous because we know we have one dominant allele, capital B, and one recessive allele in the lowercase b. All right, so go ahead and practice doing this, writing down the genotype for each of these, what are they? Chinchillas. So if we have a chinchilla that's gray, and this gray chinchilla is homozygous dominant, that means that it's going to have two capital G's in its genotype because it's homozygous. So whatever we write, the two allele possibilities have to be the same. And because it's dominant, we know that we're going to capitalize those letters. So capital G would go in this, this box here. If it's heterozygous, it's going to be capital G, lowercase g. And then finally, if it's homozygous recessive, it would be lowercase g, lowercase g. So be able to determine what the genotype is of an organism if given its phenotype, or if we tell you what the, whether it's homozygous or heterozygous. Okay, let me move my little guy. Oh, I can't move anymore. Shoot. So if we look at our genotype and phenotype ratio, remember that genotype refers to the alleles themselves. So genotype for our pea plants, so our F1 generation of our pea plants, our genotype is 1 to 2 to 1. We have one homozygous dominant genotype. We have one homozygous recessive genotype. And then we're going to have two heterozygous genotypes. Now, in contrast, the phenotype is the actual trait. So what is the physical manifestation of this genotype? So if we're homozygous dominant, this plant is going to look purple. The flower of the plant will look purple. If they're heterozygous, remember, heterozygous genotype will lead to that dominant trait being expressed. So if they're heterozygous, they're also going to display as purple. And then finally, the only time that we're going to see a white flower is if that flower's genotype is homozygous recessive. So the only case that we're going to see a white flower is if we have two lowercase p's as our genotype. So our genotype, although our genotype is 1 to 2 to 1, our phenotype is 3 to 1, right? 3 purple flowers to 1 white flower. So noisy in my neighborhood. Okay, so again, let's continue to expand on Mendel's conclusions from his pea plant experiment. And we already talked a little bit about the idea that chromosomes separate during gamete formation, but Mendel actually came up with this, at least a half century. I'm going to pause it really quick because it's noisy. Okay, sorry about that. My neighbors were being really noisy, so I wanted to just pause the video until they were done. Okay, so one of Mendel's famous conclusions was we talked about this when we talked about meiosis, but he came up with this idea at least 50 years, 75 years before they even knew what chromosomes were. And we weren't even calling them genes when he made this discovery, and he was still calling it a heritable character or a heritable trait. And he concluded that those two alleles have to separate during gamete formation. So if we think about what this means, and again, we've already gone through this in meiosis, we have a cell here that's going to undergo meiosis, or it is undergoing meiosis. And we have our duplicated homologous chromosomes, one from dad, our blue sister chromatids here, and our red sister chromatids from mom here. And these sister chromatids are in this tetrad is going to separate during metaphase 1. And that separation, remember, segregation basically means separation. And when we do this, that means that one of these will end up in one cell or one gamete, and the other one will end up in another gamete. And we know through meiosis, because this is, there are equally probable possibilities, right? All of these tetrads get shuffled every time we go through meiosis, that we have all of these different variations in which chromosome might end up in which gamete. So again, we're talking about generating that genetic diversity, and these gametes will have one allele at the end, and we already know this. Arrange this a little bit. Okay. So when we talk about this separation, we know that the alleles are going to have to separate, and we do this kind of in our Punnett square. So if we think about, we're going to cross our purple flower with our white, in our parental generation, right? Our purple flower with our white flower to get our F1 generation. And we know that our F1 generation, our hybrids, are all heterozygous. We see that right here. So when we cross our F1 generation to make our F2 generation, we're going to separate or segregate those alleles. In other words, half the gametes are going to get that dominant allele and half the gametes are going to get that recessive allele. So we can do this, and again, if you've done the lab, you're kind of well versed in this already. We can form this very simple kind of tic-tac-toe pattern, if you will, to help us predict how those alleles will meet up with the other gamete in fertilization. So if we have the plant, it looks, so here's the sperm from F1 generation, here's the egg from F1 generation. Notice that they have one dominant allele, one recessive allele, and when we cross these, our first, we're going to get 4 possibilities. So we have homozygous dominant. Let me move this guy. We're going to have homozygous dominant, 2 heterozygous possibilities, and 1 homozygous recessive possibility. Again, that 1:2:1 genotype ratio that we saw before. So a Punnett square, constructing this very simple Punnett square, helps us predict how those offspring, what the chances of those offspring having a particular trait will be. And again, remember, Mendel saw exactly this. When he bred this F2 generation, he kept seeing a 3:1 phenotype ratio of purple flowers to white flowers. So that gave him a lot of confidence that his hypothesis of segregation was correct. All right, the next conclusion that Mendel was able to make is the law of independent assortment. And so he wanted to ask if 2 different, how should I say this, alleles that represent different characters were tied to one another. So we have a parent, so now we're going to look at 2 different characters at the same time. So in this case, we're looking at, what are we looking at? Seed color. So the seed color traits could be yellow or green. And then we're going to look at the seed characteristic. It can be smooth or wrinkled. And so you can see that one of our parent generations is going to be true breeding for yellow, for the dominant traits, yellow and smooth, whereas the other parental generation is going to be true breeding homozygous, homozygous recessive for both of these traits. So this parent is going to be green and wrinkled. So we know that the only gametes that this parent can contribute are the capital Y, are the dominant traits, the capital Y and capital R. Whereas the recessive, the parent that has the recessive traits can only contribute those recessive alleles, the lowercase y and lowercase r. So now we're going to go and, so we cross our parental generation, we get our F1 generation, and we know that our F1 generation has to be heterozygous for both of these traits. And we see that here, right? Capital Y, lowercase y, capital R, lowercase r. And of course, its phenotype is going to be the dominant phenotype. So this is where his next law comes in, this law of independent assortment. And what he wanted to see is if each of these alleles, so the allele for the texture of the seed and the color of the seed, if they were dependent on one another or if they were independent of one another. In other words, are we more, do we get all of our genes randomly, or is there some type of order to how they are divided up in our gametes? And if they are dependent on one another, he would see this ratio of this phenotypic ratio of, again, 3:1, because it's going to follow that same pattern of a monohybrid cross. In other words, it's going to follow that characteristic 3:1 ratio. Now, if those alleles separate and separate themselves independently of one another, he's going to see a much more complex phenotypic ratio of 9:3 to 3 to 1. In other words, he's going to see a mixing of these phenotypes. He's going to see some yellow wrinkled seeds, he's going to see some green smooth seeds, and so on. So which one did he find? Of course, he found that they're independently assorted from one another. When he did this experiment thousands and thousands and thousands of times, he saw exactly the phenotypic ratio that he expected, that 9 to 3 to 3 to 1 ratio of these phenotypic traits. And what this allowed him to conclude is that inheritance of one trait has no effect on the inheritance of another. It truly is like we're shuffling. If our genes are like a deck of cards and we have 50, we have 52 or chromosomes, let's say. If our deck if our chromosomes are like a deck of cards, every time we shuffle that deck, we're going to get a new and random order of cards. There's no card that is like stuck to the back of another card, for example. So every time we form gametes, they're going to separate into all of those tetrads and sister chromatids are going to separate independently from one another. And it truly is random which one will actually end up fertilizing that other gamete to have fertilization. So here's a nice kind of summary that shows these two really important laws, the law of segregation and the law of independent assortment. Here through the context of meiosis. So Mendel was able to show it through physically through his pea plant experiment to show their physical traits, but we know that this actually follows very closely with the chromosome the chromosome model of inheritance, where this bears out through meiosis. So in other words, when meiosis is occurring, we know that those alleles are going to separate. So whenever we form gametes in our own bodies, we're going to only pass on one of those chromosomes per gamete. So we're literally dividing our genes in half to produce our gametes, because and again, it has to be half, because when it is fertilized, now we have a full set of chromosomes, right? If we didn't do that, our zygote, our fertilized egg, would have 2 times the amount of DNA and 2 times the amount of chromosomes that it should have, and that's just not going to work. It's not going to be successful. It's not going to be a successful fertilization. So our alleles, our genes, I should say, our chromosomes have to separate during this process of meiosis. And when they separate, that separation is random. All of the chromosomes separate independently of one another. All right, so let's review some of these concepts that we touch on in lab as well. So again, if you've taken the lab, it kind of follows really nicely with this lecture. And everything that we've talked about so far has followed a very nice model of complete dominance. In other words, when we have 2 different phenotypes, for example, purple flower and white flower, we're going to have one that's clearly dominant over the other. So we can say that even in a heterozygous genotype, like we see here and here, our phenotype will still display the dominant characteristic. So whenever we have complete dominance and we're doing that F1 cross, we're always going to see, the F1 cross to get our F2 generation, we're going to see that 3 to 1 phenotype ratio. So it's important to understand that, again, a dominant allele doesn't like screw up a recessive allele. It's just that this recessive allele is the one that fails to be shown. And it's also important to know, and I think I talked about this earlier, the dominant allele isn't necessarily more abundant than the recessive allele in a population. If that were true, we would have more people with achondroplasia than we have normal sized people. And that doesn't happen. Of course, that doesn't happen. So let's try an example here. So if we have cattle, in cattle we can have cattle with long horns or short horns. Of course, long horns is dominant. We know it's dominant because it has the capital H. And shorthorns are recessive because it has that lowercase h. And if a shorthorn bull is mated with a homozygous longhorned cow, what is the genotype and phenotype ratio of the offspring? So pause the video, try to do this one on your own and see if you agree with me. So let's move myself again here. So let's first look at the genotype here. I did the Punnett square. You can see the shorthorn bull is shorthorn bull. Yeah, the shorthorned bull is homozygous recessive, so he can only have 2 lowercase h's as an allele. So we put 1 here and 1 here. And the cow is homozygous. Yeah, homozygous dominant. So she has 2 capital H's. So we're going to put 1 H here and 1 H here. And now we're going to do our cross. So H here, lowercase h is going to give me this guy. H will give me this guy. And you can see that when we cross 2 homozygous parents, we're always going to get 4 heterozygous offspring. So our phenotype and genotype ratio is actually kind of boring for this one. Our genotype ratio is going to be 4:0 heterozygous. They're all 4. We have 4 heterozygous offspring and 0 anything else. We have no homozygous dominant. We have no homozygous recessive offspring. In other words, 100% of the offspring of this mating is going to, those offspring will be heterozygous, and therefore they'll all have the dominant phenotype, which is of course longhorn. So 100% of the offspring born to this couple will have longhorns. The ratio is 4:0. Okay, so as you know, if you've taken the lab, incomplete dominance is not the only story. That's not the only model of genetic inheritance. We can have other mechanisms of inheritance happen. And the first one that we're going to talk about is incomplete dominance. And incomplete dominance is kind of like the blending hypothesis that they had hundreds and hundreds of years ago. And in that, the phenotype of the offspring will be an intermediate between the 2 parents. In other words, we actually have a third phenotype occur. So if we have a true breeding red flower and we have a true breeding white flower, so you can see their genotypes right here. And if we cross these 2, we're not going to get a red flower or a white flower. We're going to get something in between. We're going to get a pink flower, our third phenotype. So if we look at the cross between these 2 parents, we're always going to get a heterozygous offspring in our F1 generation. Now, if we were to cross this F1 generation with itself, remember everything's heterozygous here, we're going to see... All right, we're going to see that genotype ratio show up 1 to 2 to 1. Remember that genotype ratio is different than our phenotype ratio. So our genotype ratio is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Now, the phenotype ratio is going to be different for incomplete dominance because we now have that third phenotype trait. So we're not going to just get red and white flowers. We're going to get 25% red flowers, 25% white flowers, and 50% pink flowers. So incomplete dominance, an interesting kind of variation on complete dominance. Now, remember that offspring can only have 2 alleles for a given trait, right? We only get 1 allele from our mother, 1 allele from our father, but the possibilities for what gene goes in that allele can be more than 2. A perfect example of this is our blood type. I just don't have a good spot to put this. Maybe up here? There we go. So for a perfect example of this is our blood type. So our blood type can be determined by 3 different possibilities that go in those 2 alleles that we have. So the 3 allele possibilities are the allele IA, an allele that's represented by IB, and then finally an allele that is represented by lowercase I. And what these do is they represent, so these will these code for a gene that will end up putting a very specific type of carbohydrate on the outside of our red blood cells. And we can get any combination of these three alleles, but we can only get two of them. So let's look at how that works. So here are all of the possibilities of our of those multiple alleles. So let's just focus on this row right now. So an individual can be IA. They can be IA lowercase I. They can be IB, or IB lowercase I. Next, they can be IAIB. And then finally, they can be lowercase I. In other words, these 1, 2, 3, 4, 5, 6 possibilities for our genotype for blood type. All right, so we have these 6 different possibilities: 1, 2, 3, 4, 5, and 6. But now we want to I want to talk about another variation on dominance. Remember, we have complete dominance, we have incomplete dominance, and now we have codominance. So codominance is when we have 2 different alleles. So if we have 2 different alleles being expressed, they're going to affect the phenotype, but in 2 distinguishable ways. In other words, there's no blending. So in incomplete dominance, let's just go back to this. We had a true blending of the red and the white to end up with this pink phenotype. So we actually had a third phenotype occur. Whereas in codominance, what's going to happen is if we have a heterozygous individual, we could have both of those traits being expressed independently of each other at the same time. So let's look at what happens here. So I know this is a little confusing, but let's look at the actual phenotype of our red blood cells. So if we have these red circles here represent our red blood cells. And remember, if we have the IA allele present, we're going to have this triangle carbohydrate on the outside of our red blood cells. Whereas if we have a B allele, IB allele in our genotype, we're going to have this circular carbohydrate expressed on the outside of our red blood cell. Here's where the codominance comes into play. It's possible that an individual could have one allele that represents IA and one allele that represents IB. And in this case, neither one is dominant or recessive to another, nor do they blend. Right? So we don't have complete dominance and we don't have incomplete dominance. We simply have codominance. In other words, both the triangles and the circles will be expressed on the surface of this red blood cell. And then finally, we could have a situation where we have both of the recessive traits, the lowercase I trait, and in that case, we're not going to have any carbohydrate expressed in our red blood cell, and we're considered a type O. So you can see the phenotype for each possibility here. There's 2 genotypes that could lead to the A phenotype. There's 2 genotypes that would lead to the B phenotype, type A blood or type B blood, and so on. There's only 1 genotype that would lead to being AB, and there's only 1 genotype that would lead to type O. And as you can imagine, blood type A and blood type B are by far the most common blood types that we see in a population, because there's more genotypes that would lead to that phenotype. All right, so the next concept, again, the next kind of variation on complete dominance is the idea that a trait can have many, many genes that contribute to its phenotypic appearance. In other words, there are, a perfect example of this, of course, is skin color. So there are, oh my goodness, I forget. I want to say like at least, I remember reading an article that said there's about 14 different genes currently that will affect the color and tone of your skin. Remember that skin isn't necessarily based on just the amount of melanin. It also has to do with the amount of keratin. carotenoids in your skin color. There's other pigments that contribute to skin color, for example. And these genes can basically exist along a continuum. So you can imagine if we have, so here we're doing 1, 2, 3, 4, 5, 6. So here we have 8 different alleles that can contribute to skin color. And if we get one of those 8 possibilities from our father and one of those 8 possibilities from our mother, that means that there's 64 different possibilities for skin color that you'll end up as your actual phenotype trait, right? So when you see the color of your skin, it's being dictated by how should I say this? A potential combination of 64 different genotype combinations. So this is polygenic, and I would argue, I'm not a geneticist, but I would argue that most of our traits are polygenic. There's a very, very complex interplay between genes and how those genes manifest as phenotypic characteristics. And then I would also take that a step further and say that it's not just our genes and how they interact with one another, it's also how our genes interact with the environment in which we develop in. Of course, I mentioned in the previous lecture about there's different growth different conditions under which an embryo develops in the womb, and things that the mother is doing, the types of hormones that she's expressing, the type of chemicals that are present in her body can affect genetic expression. So the type of experiences that we go through in our developmental stages as young babies and children can also affect the expression of those genes. So it's not just our genes interplaying with one another, it's also our genes and how they are being affected by our environment as well. Next we have pleiotropy. So pleiotropy is a mechanism of inheritance where we have a gene that can have multiple phenotypic effects. So even though we only have one gene, and we have one gene that can change, that change in that gene can have various outcomes. A perfect example of this is sickle cell anemia. So sickle cell is a very painful disease, often fatal disease, that many people are carriers for it. So in other words, they're heterozygous for this sickle cell allele. And when you are recessive, you will get sickle cell anemia. And what happens here is the hemoglobin in our red blood cells will not form correctly. It will become misshapen, and that causes our red blood cells, instead of to look like a nice fat little donut, it looks like a crescent. It kind of has this sickle shape to it. And that causes all kinds of problems. And you can see the various phenotypic downstream effects of this, of having this particular allele. So what happens to these sickle-shaped cells is that the red blood cells break down. That makes the individual that has it basically always be anemic, right? Their red blood cells are dying at a much faster rate than a normal person, and that anemia will lead to physical weaknesses. It also causes those odd-shaped red blood cells to get clogged in capillaries, and that can lead eventually to heart failure. It's extremely painful. Your joints are very painful, and sometimes just the pain itself can be damaging. It's a very, very difficult disease, very painful disease to live with, and then it can also lead to damage in our secondary lymphoid organs. So our immune system tries to clear these sickle cells. They usually tend to accumulate in our spleen and some of our other secondary lymph organs, and that can lead to spleen damage. So one allele can have various phenotypic effects based on the presence of that one particular gene. All right, I think this is my last one. This is the last variation on complete dominance, and that is that we can have a variation on inheritance based. No idea what that was. Okay, we can have variation on inheritance based on whether or not we inherit an X or a Y chromosome. remember that we will always get an X chromosome from our mother. So both male and females receive an X chromosome, whereas so the egg will always contribute an X chromosome, whereas the sperm is the one that will actually determine the gender, the sex of the child, in that if it contributes an X chromosome, the sex of the offspring will be female, and if it contributes a Y chromosome, the sex of the offspring will be male. So what happens here is we can have, what this is important because remember that Y chromosome is very small. So the X chromosome is a full-size chromosome and contains the complete number of genes, some number of genes on this chromosome. Whereas because that Y chromosome is much smaller, it doesn't carry the same amount of genes. It has a lot of genes that are missing. So if the father has is unaffected by whatever this particular trait is, but the mother is a carrier, when they have offspring, if you look at their female offspring, they're not going to be affected because the father can only contribute his normal X chromosome, while the mother can contribute either the normal X chromosome or the trait, the chromosome that carries this particular trait, the affected chromosome. So a daughter can either be unaffected, so she would have both copies of the normal X chromosome, or she could be a carrier, because remember, this is a recessive trait. So even though she has that trait to be affected by whatever this particular disorder or trait might be, she has that dominant allele to kind of protect her phenotype from manifesting in that whatever this disease or trait might be. But now let's look at the son. So the mother, again, so the father can't contribute a normal allele to his sons. He can only contribute that Y chromosome to his son. So if we look at the son, right, the dad is going to give them a Y chromosome. So now the boys basically have a 50-50 chance of getting one of these X chromosomes from their mother. And so in the case, if the boy has one dominant, unaffected allele, this offspring will be fine. However, if this son has it, even though this trait is recessive, he doesn't have that corresponding allele on the Y chromosome to kind of protect him from the phenotype of that outcome. So whenever we see a sex-linked trait, male offspring will have basically a 50-50 chance of receiving or having that inherited trait, while the females have a much lower chance of having that particular trait. Let's look at an example. So one example is color blindness. If you can't see the number, number 7, if you can't see the number 7 in this circle, that means you're colorblind. Colorblindness is a perfect example of a sex-linked trait. And it's much, much more common in males than females, because it's a sex-linked trait. Another example is basically hairy ears. It's kind of gross, but this is actually a genetically inherited sex-linked disorder. So you can see this actually ran in the royal family in England, Queen Victoria and Prince Albert. If you ever have a chance to go to London, go to the Victoria and Albert Museum. It's amazing. But Queen Victoria was a carrier of for this hair, basically. Hair, I don't know why I'm laughing. I'm such a juvenile. But for hairy ears. And so you can see that she passed that gene along to her daughter Alice, but because Alice had a normal X chromosome from her father, she was just a carrier. She did not have the phenotype of having a hairy ears. So Alice passed it along to her daughter Alexandra, and then when Alexandra married Tsar Nicholas of Russia, when they had a son, their son Alexis, even though he was I shouldn't say even though. So even though he only had one copy of this recessive allele, that Y chromosome didn't protect him from having this. I guess it's not really a disorder. It's not that big a deal. But he had the phenotype of having the hairy auricle because his Y chromosome didn't protect him from that recessive allele in this particular case. So more bad things happen to this family, but this is a science class and not a history class, so you can investigate that on your own. I think that does it. Is that, yeah, great. So that's our last slide for this part of the chapter, and also for chapter 9. So I will see you in chapter 10.